Okay, so we're going to start talking about a protein called hemoglobin, and also we'll talk about another protein called myoglobin. So these are two of the most studied proteins that are usually used as models when we talk about protein binding and structure and function. So we're going to see what's happening here, how the structures of these proteins are impacting the role that these proteins have, what their job is. So first, let's kind of talk a little bit about what myoglobin and hemoglobin are. So myoglobin and hemoglobin are both oxygen binding proteins, and I'm going to talk a little bit about what their similarities and differences are. So let's start with myoglobin. So again, anytime you can look at the name and learn something about it, that is always helpful. Myo means muscle. Globin means it's a globular protein, just like immunoglobulin was an immune system globular protein. So now myoglobin, myo means muscle, globin means this is a globular protein. So myoglobin is a globular protein that has one subunit. So we haven't used that word subunit before, but this is tertiary structure. We're going to see the difference between tertiary structure and quaternary structure here in a second. So it has one subunit. It has one oxygen binding site. One place to bind one oxygen molecule. Myoglobin binds oxygen quickly and strongly and does not release oxygen until the partial pressure of oxygen is very low. Myoglobin is found in the skeletal muscles, and myoglobin, its major role is storing oxygen. So now we're going to look at the similarities and differences between myoglobin and hemoglobin. Heme means the heme molecule. These are in red blood cells. And globin, again, this is a globular protein. Hemoglobin has 4 subunits. This is quaternary structure. So we've said that proteins have 4 levels of structure: primary, secondary, tertiary, quaternary. What quaternary structure is it's just multiple subunits. Sometimes you'll see the word domain instead of a subunit. It's multiple subunits or domains of tertiary structure. So myoglobin has 1 subunit. Hemoglobin, because it has more than 1 subunit, is labeled as quaternary structure. Because it has 4 subunits, you might also see hemoglobin is referred to as a tetramer, tetra for 4, a tetramer of subunits. These 4 subunits each have 1 binding site, so hemoglobin has 4 oxygen binding sites. Hemoglobin binds oxygen less strongly, and it releases oxygen more readily at a higher partial pressure of oxygen. Hemoglobin is in our red blood cells. And hemoglobin is good for transporting oxygen. So hemoglobin and myoglobin are both oxygen binding proteins. I'm not going to draw out all of the structures. They all are labeled on the PowerPoint slides. All of this about hemoglobin and myoglobin starts on slide number 23 of chapter 7, and it's all drawn out, and I'm not going to draw all of the pictures of the structures of hemoglobin and myoglobin, but I want you to understand the difference between these two proteins. Hemoglobin is essentially 4 subunits of myoglobin. It's not exactly the same, but it is very similar. Hemoglobin, its job is to transport oxygen. Hemoglobin is in our red blood cells. Our red blood cells are essentially just full of hemoglobin. That's all they are. They start out as something called reticulocytes, which have a nucleus and hemoglobin, and then as the red blood cell becomes mature, the nucleus is ejected, and now it is an erythrocyte, which is the only cell in our body that has no nucleus, because once it becomes mature, the nucleus is ejected. So the reticulocyte is the immature red blood cell that still has a nucleus. Once the red blood cell is mature, it ejects the nucleus, becomes an erythrocyte, which is a red blood cell. And the only thing that red blood cells do is they transport hemoglobin. Red blood cells kind of are what's called biconcave. It kind of looks like a donut or a bagel with no hole in the middle, but like a dip in both sides. And that dip in the middle is to increase the surface area. The more surface area the red blood cell has, the more oxygen these red blood cells can transport. So what happens is hemoglobin in our red blood cells picks up oxygen in the lungs. It picks up oxygen in the lungs and it transports it throughout the body. Or it can store it in the skeletal muscle. And it is stored in the skeletal muscle by myoglobin. So it can transport the oxygen from the hemoglobin to a myoglobin, which will then hold the oxygen and store it in the skeletal muscle. The myoglobin in the skeletal muscle is holding on to the oxygen and saving it, so in times of extreme exercise or really low oxygen levels, then it releases it. The purpose of hemoglobin is to pick up oxygen, take it somewhere, drop it off. Pick it up, drop it off. It's not meant to store oxygen. The purpose of myoglobin is to pick up oxygen and hold it until there is a time of severe oxygen depletion and then release it as kind of a backup. So even though these proteins are shaped almost exactly the same, this one has 4 times as many subunits, they have very different jobs. And the reason for that is because when you have more than one subunit, these multiple subunits allow for something called cooperative binding. So hemoglobin has cooperative binding, myoglobin has non-cooperative binding. So to have cooperative binding, you have to have more than one subunit, more than one domain. So this cooperative binding exists in 2 states. One state is called the T state for tense, and one state is called the R state for relaxed. The T state is deoxyhemoglobin. HB is the abbreviation for hemoglobin. It's deoxy, it has low affinity for hemoglobin. The R state is what's called oxyhemoglobin. This is hemoglobin that is carrying the oxygen. It has a high affinity for oxygen. Sorry, that was supposed to be an oxygen. So this is all constantly happening as we're going between the lungs in different places. So the affinity for oxygen is high in the lungs and low in the extremities as we're releasing the oxygen. So we would have our state, these are conformational changes, two conformational possibilities. And when you look at the slides, you'll see the shape of the hemoglobin kind of flexes a little bit between this T state and R state. And when you have this movement between these four subunits, it opens and closes the pockets and the location for the oxygen to bind inside that hemoglobin. So this is cooperative binding, and we have cooperative binding when we have more than one subunit, and then that impacts how binding is happening. So what happens with cooperative binding is that binding at one site is going to increase affinity for all the other sites. So I'm just going to represent the 4 binding sites for hemoglobin as this square with 4 pieces. So what happens in cooperative binding is if oxygen binds here, then that means it then increases the affinity for oxygen at the other 3 sites. And the opposite is also true when we go to the T state. If you lose the oxygen at one site, it decreases the affinity for oxygen at the other 3 sites. So this is called cooperative binding, where binding at one site influences the binding at the other sites. And this is for all proteins with multiple subunits. This isn't something that's specific to just hemoglobin. So let's look at something called a binding curve. And we'll look at the binding curve and compare myoglobin and hemoglobin. So this is in your textbook and in your slides, but I'm going to draw it out and we'll talk about it a little bit and see what is happening throughout the binding of oxygen. So we're looking at the oxygen binding curves for hemoglobin and myoglobin. So what goes on the y-axis is something called YO2. This is the fractional saturation of oxygen. And then down here on the x-axis is PO2. This is the partial pressure of oxygen. So there's 2 important parts on this partial pressure of oxygen axis. Somewhere down here around like 37, we'll say. Let's just say 20, 40, 60, 80, 100. So we'll say down here around about 37 millimeters of mercury. This is the partial pressure in the lungs. And then up here around 100, this is the partial, sorry, I said that backwards. This is the partial pressure down here in tissue, like skeletal muscle. And this up here around 100, this is the partial pressure in the lungs. So the partial pressure in the lungs, this is where the hemoglobin is picking up oxygen. And then in the tissue, this is where it's releasing oxygen. So hemoglobin, let's do hemoglobin first. Hemoglobin has what's called a sigmoidal curve. It kind of looks a little bit like an S. This is the sigmoidal curve. This is hemoglobin. This is the binding of hemoglobin. Myoglobin, this sigmoidal curve is representative of cooperative binding. Myoglobin does not have a sigmoidal curve because it is non-cooperative. The myoglobin binding curve looks something like this. And the abbreviation for myoglobin is MB. So myoglobin and hemoglobin bind oxygen differently because of this cooperative binding. So in the lungs, you want hemoglobin to have a high affinity, a high fractional saturation of oxygen. And then in the tissue, when the partial pressure drops, we want it to have a low affinity because we want hemoglobin to pick up oxygen in the lungs and we want it to deliver in the tissue. And this is the whole point of this cooperative binding. So up here, this would be the R state where it's relaxed. This is where we have oxygen. So I'll say this is oxyhemoglobin. And then down here in the tissue, this would be the T state. This would be deoxyhemoglobin. Because we want the hemoglobin to pick up the oxygen in the lungs and we want it to drop it off in the tissue. So this affinity for oxygen has to do with this conformational change in the hemoglobin. And this conformational change in the hemoglobin brings this iron, so heme comes from the word, from iron, and it changes, when it changes the conformation, it brings this iron that's in the middle of this protoporphyrin ring. You can see all of this in the pictures, but the oxyhemoglobin is going to bring the iron in the plane of the ring. Protoporphyrin ring. This will help it bind the oxygen. It can only bind the oxygen when the iron is in the correct conformation. That's what the oxygen is binding to, is to this iron. The deoxyhemoglobin, when we have this conformational change, the iron is now below the plane of the ring. So now it cannot bind oxygen. So this is also represented here. The conformational change of this cooperative binding between these two states, T state and R state, will allow oxygen to bind here and release the oxygen here.